Hey everybody, I'm Sherry Seligson. I'm an apologia author of science and other curriculum, and we are going to continue our series of doing just some fun activities. They are kind of science related because I love science, but I'm going to try to pull those topics into other subjects as we go. We've been doing this for about a week and a half now, and it's just been really fun. I'm trying to get on every day at around 11 o'clock. Taking off Sundays just for some rest, and maybe Saturdays too. But we're going to do some really fun stuff as we progress through this. Now, what we're going to do today is kind of based off of yesterday. Yesterday we talked a little bit about what viruses were and how our bodies fight them, how they're not living organisms, and how that's why antibiotics don't work. If you don't remember what there's different criteria for life, and viruses don't meet one of those especially. And one of those main criteria for life is that viruses can't reproduce by themselves. They actually take over a cell and they insert their own instructional DNA into that cell and tell the cell, make more of my stuff, not your stuff. And that's not good for cells, that's not good for the organism. And so that's why antibiotics don't work. But I didn't say this yesterday, and I want to inspire some of you students that we didn't know that there were natural antibiotics that existed years ago. Because pathogenic bacteria, which are living things that can harm people and other organisms, exist in the world, then of course it would make sense that there were natural ways that other organisms created their own antibiotics. And sure enough, that happened when we discovered that antibiotics could be produced, for example, penicillin, from a penicillium bread mold. And there are other organisms in creation that have natural antibiotic abilities, antibiotic chemicals that fight off living bacteria. Wouldn't it be more obvious then that there perhaps would be organisms that have antiviral chemicals that they produce? We haven't discovered them yet, but maybe one day one of you guys will. So hopefully we'll have, rather than antibiotics, we might have antivirotics, I guess. I don't know. But it may be out there, we just haven't discovered it yet. And I wanted to talk a little bit about more what DNA is, because a virus has a protein coat around it and then just a little bit of DNA or RNA, which are two instructional chemicals that tell a cell what to do. And I like to use the example of a Lego kit. You get your instructions, you have all the pieces, and you need to know how to put those pieces together. Well, that's what DNA does. It's a shortened acronym for deoxyribonucleic acid. A very long molecule, very big. It's coiled up really, really tiny and compacted tightly into the middle of cells, but it tells the cell, make this part, make a wheel, make this, and then put it together this way, put it together this way. And so the cell is instructed to do that, and that's how the cell knows what to do, by the instructions of the DNA. So we are going to find some DNA within the cells of an onion today. We're going to make our own little model of DNA with some paper. So we're going to, midway through this little process that looks all chemically here, we're going to stop, let it sit for a minute, and make our own DNA model. Now, DNA is so huge, it has to be able to be tightly packed into cells. And so it's packed into the center of the cell called the nucleus and wound around spools and spools of proteins. In fact, if you were to unspool all of the DNA in just one cell, one human cell, it would be over 2 meters long. That's taller than me, okay? 2 meters long, packed into something that we can't even see without a microscope. So let's go ahead and get started and see if we can isolate some DNA. Now, I'm gonna call this my quarantine lab, okay? We're doing quarantine experiments here because I don't have access to a lot of things. I don't wanna run to the store every day and pick up stuff because obviously I don't wanna be spreading a lot of things or catching something. And so I'm using what I have, and one of the main things I don't have today is meat tenderizer, but we're gonna try to see if we can improvise. And I also normally do this experiment with split peas, but you can do it also with onions. So we're gonna start by taking our onion, and I'm gonna cut it up into some chunks and stick it in a blender. And interestingly, if you have access to a microscope, you can peel inside the onion. There's an onion skin, like in between each layer of onion. Oh, it's gonna make me cry. Oh. And you can see this skin. This is something that you could look at under the microscope and actually see the individual cells because it's very, very thin. And so if you have access to a microscope, you can do that. I might see if I can try to do that later today and take a photo. If I can, I will upload that in the comments of this video. So we're gonna put this in, and then I'm gonna take a cup of regular water, and I'm going to add I'm gonna add a 1/4 teaspoon of salt to that water. We're gonna make a salt water substance. And then I'm gonna pour it into this glass so I can stir it up. I wanted to measure out that cup of water. Actually, I'm gonna get some of that salt back in here. Swirl it around. There we go. And then I just want to stir it around to make kind of a saltwater solution. What we need to do is we need to get into the cells because DNA is located in the very center of cells. So we have to do a series of chemical processes to get into the center. So this first part is to mechanically get in. We want to break up our onion cell in a way that we can get the individual cells kind of floating around in solution. All right, so my salt has dissolved. I'm going to pour it into a blender. You know, I had better vision. I'd be able to read what you guys are saying. Everybody's saying something. Anyway, I hope you're enjoying it. So we're going to go ahead and whirl this around for just about 20 seconds or so and break it up. Kind of onion stew, but not cooked. All right, so we have our little onion mix here, and we're going to strain it out. We want just the liquid material. So I have a glass, just a glass right here, and I'm gonna put a strainer on top of the glass, and I'm gonna slowly pour this into the strainer just to get out the big chunks. There we go. And so what we have in the glass container now, hopefully, is some fluid, some water, some salt water, with some chunks or tiny bits of onion cells. Now, incidentally, this lab is one of the labs that's in one of the Apologia science texts. General biology has this. But it's fun because you can do, believe it or not, you can actually see DNA using simple household materials for the most part, without fancy science glassware or whatever. And we try to do that because we really want you to be able to do science at home and have fun with this. Ask those questions and get going. Okay, so I've got a good job of separating my onion juice, if you will. And so now that we've got that, we're gonna add some soap. So this broke the cells apart. Now we're gonna add some soap. We want 2 tablespoons of liquid hand soap. And you want to use clear hand soap if possible, not the foaming kind, but this is concentrated enough that what this will do is it actually dissolves and breaks up the membrane surrounding the cells, and so it makes them pop. And so what you'll end up getting is, after a while, is the cells will open up and just kind of dump all of their contents into this liquid, and that's what we're wanting to happen. So I'm going to add the soap. I'm not going to whip this up because if I do, I'll get foam all over the place. I'm just going to stir it around in a gentle swirl to distribute that soap. And we want this to sit for about 20 minutes. I'm sorry, 10 minutes, not 20. That would take too long. And so while we let this sit, so I have my little soapy material here with my onion cells. I'm going to let that sit. And we're going to do our little craft. We're going to make a model of DNA. Now, there's lots of DNA models out there. Let me push this. I'm trying to work within the little box of my phone. So bear with me on this one. Okay, so I have some paper here. And our goal is to build a basic structure of DNA. Scientists have experimented and done several different tests to determine the shape of DNA, deoxyribonucleic acid. It actually has 2 backbones, if you will, and then connectors made from nucleotides. And those things connect like a ladder. So we're going to make a little paper ladder. And actually DNA is a little bit more than this, but we'll do that in a second. So what you need to do to make this is you need 2 sheets of 8.5 by 11 paper. You can actually do this with newspaper if you have newspaper anymore. And I'm going to cut mine a little narrower. I'm going to cut maybe about 2 inches off of both of my sheets. Remember, we're letting our DNA cook right now. All right, so this is an activity to do with all ages. So here we go. This is, these are my 2 sheets, and we're gonna take 1 sheet and we're gonna start rolling it up into a tube. And I'll show you, can you guys see that? Yeah, I think you can see that. Okay, so I'm rolling it up into a tube, just in case, just nothing fancy. And I wanna make it a little narrower, so I'm gonna stick my finger in there and twist it. You and the kids can do this and have fun. Actually, paper folding is a really fun activity, or paper modeling. Okay, so I have a little tab sticking out. I'm gonna take my 2nd sheet and stick it in here, just like that, and keep folding. You can do this for multiple sheets if you want, but I'm just gonna do 2. The more you do, actually, the bigger your ladder can get. I wanna tack this little flap right here. I'm gonna tack it down with just a couple pieces of tape so it gets kind of fiddly if it gets really long. So just 1 on each end, 1 here, 1 here, and then we'll just keep rolling. That's the motto of this, these last couple of weeks. Keep on rolling, right? We can do this. We are here to help you guys pass the time productively and have fun. I'm always available to have a conversation if you guys want. If you got your kids at home and you're like, what do we do with my kids? I'm happy to talk to you about that. I've homeschooled my 4 for 21 years. We just finished our last one a couple years ago, but I still love talking about science and education. I've got my master's in education, and I'm a marine biologist actually by trade. So, okay, we have our little cigar, if you will, and we're gonna just tape both sides together like this. Again, this is so it won't, you don't have to use tape, but I like to, just so that it doesn't fall apart. Okay, and then we're gonna make a cut in 2 spots, maybe a third of the way in from each end. And we're gonna cut down, not all the way. So I'm gonna take some scissors. You gotta kind of have to smash it like that. You see what I'm doing right there. And I'm gonna cut down, and you don't wanna cut it all the way off, but you wanna cut down towards the bottom, and I'll show you. You want to leave a good, maybe a 1/4 inch of connection, so you can kind of see how far I've cut to that point. Okay, I'm gonna do it again on this side. This might be a mom and dad thing, depending on how old the kids are. Okay, so I have my 2 cuts, and we're just going to reform it into a tube shape, and then bend each side down like that. Okay, that's all you're doing. And then we're gonna cut across the top here, just 1. We're gonna stick our scissors like this into the tube and cut across the top of this middle piece. If you use lots and lots of strands, it might be hard to cut. That's why using maybe tissue paper or newspaper might be easier to do. Okay, so now we have this funny little shape. Because this is all spooled up, you can actually start to push with your finger this way and pull with your finger this way to get it to start coming out. Can you see? I'm working on it. I'm going to try to separate these because they're kind of attached together from me cutting. And I'm going to start pulling and pushing and pushing. Can you see it coming out a little bit more? All right, now once you get it started a little bit, it becomes a little easier. And you can grab just a couple of the top pieces and start to pull and push. There we go. There we go. Takes a little fiddling. But can you see I'm starting to get a ladder? Don't want to do it too tightly. There we go. Don't pull it too much because you'll separate your layers out. Okay, so the goal is, it's kind of a wonky little looking ladder there, but let's see if I can get some of the top ones here to work. They're not wanting to come out. But the point of this is we're making a DNA structure, and you can actually look up, there we go, you can look up online the actual chemicals involved in this. These are made up of molecules attached. There's our nice little ladder, you see that? And interestingly, DNA is a ladder that is twisted like this, and it spirals around itself. It's two spirals, and that's called a double helix. And so that's what our DNA model could look like. That's what it actually looks like. And based on experimentation, scientists have actually figured out that's what it does. Now, even the way it unzips itself down the middle and replicates, RNA, you may have heard the term RNA, which is ribonucleic acid, is pretty much just 1/2 of this molecule, just one side. If I were to cut it straight down like this, it's 1/2 rather than both. And it has a lot of important jobs within a cell too. And some viruses have RNA only, some viruses have DNA. And so we all have DNA in our bodies, and it's a very important molecule for telling our cells what to do. So this is one way you can make a DNA model. There we go, our double helix structure with the little nucleotides in the middle. I'm trying to give you guys some vocabulary words. Okay, so now that we've done that, this was kind of fun. Let's see how we're doing with our isolation of DNA out of our onion cells. Let me pull this closer in. Okay, so I've been letting it sit in the soapy solution so that the soap molecules, we talked about soap yesterday and how it's a very big molecule, and it has one side that's polar and one side that's non-polar. And the non-polar side likes to dissolve with non-polar materials like fats and things. The polar side will dissolve with polar materials like water, and so it helps connect the fats and the oils on our skin so we can wash that stuff away from our skin. Well, the same thing within cells. Cells have polar molecules and non-polar molecules, and those connect with the soap. The soap pops the cell open, all the cell contents pour out, and so hopefully that's what's happened right here. They've opened up. Now the next step, we want to get into the DNA molecules. Remember how DNA molecules are so tightly packed and spooled around each other and woven and packed in there because there's so much information that has to be in a cell. It is much more than the information in a Lego kit, right? Tons and tons of information packed into microscopic size. And so it's all spooled around proteins, and we want to break down those proteins so that the DNA will come unspool. Now, one of the ways to do that is to use meat tenderizer. Guess what? I don't have meat tenderizer, and I can't go to the store to do that right now. So we're going to improvise and use vinegar, because I know that vinegar is a mild acid and it breaks down protein. So like if you're going to marinate meat for nice steak for dinner, you might use balsamic vinegar or some other kind of vinegar solution. That's because the vinegar breaks down the protein molecules. And so I'm going to pour that vinegar in, and we're going to gently stir this. Let me use my spoon and just gently stir this, because I'm hoping that it will break up. There we go. I'm starting to see some positive things happening here. Hopefully it works. We want to unspool those DNA molecules. Believe it or not, I think that's DNA, but I'll show you in a second. Okay, and now that we've done this, we want to add rubbing alcohol, because rubbing alcohol will actually separate out the DNA. We're going to slowly add some rubbing alcohol, and hopefully it will pull up, the DNA will float up with the alcohol. So let's see if we can get that to work. Let's hope this works. I'm going to try not to disturb the surface, so I'm going to pour it alongside. All right. And let's see what we got. Okay, doesn't look like much, does it? But let me use a toothpick here. And I'm gonna swirl it around. And you know what? You guys, this is amazing. Let me use a spoon. I've got tons. I don't know if I can get it to stick. There are little white strands in here. These strands are strands of DNA, and I'm trying to spool them around and wrap them around my toothpick, and it's not working, and they're not wanting to get on my spoon. Okay, I'm gonna move the phone so you can see it. Let's see if we can grab this together and give you guys a close-up, because I don't want to tip the phone. All right, you guys got to look at this. Do you see the white material? Let me move it around. It's the strands. You see those strands? They look like strings. Can you guys see that? Can you see that? Is that not amazing? Those are strands of DNA. So we actually compiled all that DNA together, so it was packed and spooled together, and that way we can see it. It's big enough for us to be visible. So I hope that this was encouraging to you guys and something fun to watch with your kids and talk about. If you have any more any questions about this, I'm seeing there's lots of comments, so I'm going to come back and see if I can answer all the comments the rest of the day today. And we're going to do some more stuff tomorrow and the rest of this week, Lord willing, if we can come back. I'm trying to see if we can get outside a little bit and do some nature walks together and identify stuff. We did a bunch of stuff on my page last week. Sherry Seligson, author, we did something every day, and it was kind of more botany related, and we did some walks outside too, but there's lots of stuff we can see. We're going to do some physical science stuff and maybe some gravity stuff. So lots of fun things we can do that are educational, that are fun, that don't feel like they have to be something we have to do, but something that just redeems the time that we are together with our kids, to give them something to look forward to each day too. So, and hopefully give you guys something to look forward to each day as well. So thank you for joining me. I will see you this time tomorrow at 11 o'clock Eastern. And again, leave any comments in the comment section here. Thanks a lot.